So this is the second section where we're looking at the slides. I will share my screen. Okay, share my desktop. Here we go. So we'll go and look at this one here. If it does its thing. Okay. All right. So tell me, everyone, I think the diagnosis here is pretty straightforward, partly because it's in the URL. What troubles did you have with this one? You all recognize that this was a Malt lymphoma?
Diane, how do we know which tissue it is?
Okay, let's start at the beginning, shall we? We can see an epithelial surface here.
Can we go a bit closer? Yeah, there we go.
Yeah. Okay, and the epithelial surface has got goblet cells and it's got squamous cells in it, so we know that this is conjunctiva. So this is a conjunctival biopsy and it's got this infiltrate of cells in it. And the cells are kind of taking over the epithelium on this side. They're infiltrating it. As we look around, we can kind of see it's mostly a C, but in places there are kind of these poorly defined nodules. Yeah. So looking around again, the nodules really are poorly defined. So one of the tricks to realize is that in lymphoma, the nodules that form, if this is going to be a nodular lymphoma, follicular lymphoma, rather than a reactive process. So they're poorly defined, they lack polarity, they don't have those changeable bodies in them. Yeah. And the cells in them are often part of a monomorphic population. And when we stain them up, of course, we get a clonal proliferation. And when we do genetic studies, we get abnormal gene translocations. So at lower power, when I was looking at this, what I could see really was this, like, it's a C, isn't it? It's flat and it appears monomorphic, though there are in places some vague nodules. Yeah. The other thing that is important is this surface here. So in Malt lymphomas, you get lymphoepithelial lesions where the lymphocytes infiltrate the epithelium. Let's have a look at the actual cells themselves. So what we've got is a sea of little cells, and the cells are small and uniform, and I can't go any closer to them, but if I could, they would have slightly irregular nuclei. Now there are some odd, bigger looking cells here. Amanda, do you want to, how good's your lymphoma work, Amanda?
Oh, well, I can sort of see fuzzy little cells there. Now they could either be like a centroblast or they could be a histiocyte nuclei. That one looks like a centroblast.
That's right. So sometimes in lymphomas, all the cells aren't necessarily the same. You can have a mixture of centrocytes, which are these little dark ones, and centroblasts. So don't be put off by the occasional bigger cell. In high-grade lymphomas, every single cell looks big like this. Okay.
So Diane, how do you know this is high-grade or low-grade?
So I know that this is low-grade because the majority of cells are small rather than large.
How do you know they're small?
Good question. So I often compare them to the size of a red cell. And a small cell, a small lymphoma cell has only got a nucleus up to 2 times the size of a red cell. But you can see that these centroblast cells have got a nuclei about 4 times the size of a red cell. Now, Amanda's really great at cytology, so she knows a lot more about the cells than me. So do you want to help answer that question, Amanda?
Oh, well, you can, yes, you must use a known standard size. I like to use a histiocyte nucleus. There's probably some in here. If it's smaller than a histiocyte nucleus, it's a small cell it's a small lymphoma cell, and if it's larger, it's large. But there are other clues besides the size, because typically small cell lymphomas are low grade. And does anybody see a mitotic figure in this field? No, there are no mitoses. If this was a high grade lymphoma, there would be mitoses and apoptotic bodies everywhere. And remember, Diane showed you in her presentation PowerPoint at the last session, pictures of what happens when there's a high turnover rate. We have macrophages that gobble up dead and dying apoptotic cells. So high mitoses, high apoptosis indicates a high proliferative index, which we'd find in a high-grade lymphoma, such as a diffuse large B.
So I think in summary, in answering it to the rubric that we have, the normal tissues present, we've got some normal conjunctiva, haven't we? And we've got some normal conjunctival stroma. Yeah. And the main abnormality is an infiltrate of lymphocytes in the stroma and in the surface epithelium that is uniform, that is composed of small cells with only occasional mitoses, that there isn't apoptosis, and that will tell you it's a low-grade lymphoma. But I would also comment on the features that aren't there that would indicate that it's not a reactive change. So there's no mixed cell population, there's no reactive follicles with polarization and tingible body macrophages. The differential diagnosis is between a Malt lymphoma and a follicular lymphoma, and we can't tell the difference between these until we've used our ancillary tests, which include flow cytometry. So these are the extra tests that we would do. Flow cytometry. We would use genetics. We'd want to have a karyotype or FISH to detect any, probably FISH actually, to detect any chromosomal abnormalities. And we would want to use immunohistochemistry. So immunohistochemistry is going to prove that this is clonal, and it's also going to detect any abnormal genes that are being expressed, like BCL2, and it's also going to, nowadays, detect important mutations that might be present. Have you got anything to add, Amanda?
Probably the immunohistochemistry, we want to confirm that this is a B cell proliferation, because most ocular lymphomas are B cell lymphomas and most T cell proliferations are reactive. Samuel's asked a good question here. How do you know the difference between a lymphoma and reactive lymphoid hyperplasia? Because macroscopically, they'd both look like this thickened whitish salmon patch, wouldn't they?
They would. So reactive lymphoid hyperplasia will have those very characteristic reactive lymphoid follicles in them. That's the low power thing. So at low power, you see the reactive lymphoid follicles. At higher power, you can see that it's got several cell types, but in an organized way within the follicle. So it moves from centroblasts to centrocytes, as I showed you last week. There will be other types of reactive tissue present. So they'll probably maybe mixed in plasma cells particularly, or eosinophils if it's got an allergic component like giant papillary conjunctivitis. If you, and then you would go on, and if you're still unsure, do the rest of the tests. You would show that it was a mixed B T cell population. You would show that it was not clonal for kappa or lambda, and we probably wouldn't go on and do FISH after that, but if we were uncertain, we could do that. Have you got anything to add, Amanda, to that?
I would say zoom out a little bit further and look at low power. And okay, I don't see any lymphoid follicles. So at this power, Sam, I don't think it's reactive. It looks like a sea, just a complete diffuse sea of all of the same, except for, and it's going into the epithelium. I don't see any follicles. So if you Google up what do lymphoid follicles look like, there aren't any. So it looks like it's a neoplastic proliferation because it all looks the same to me.
Okay, and
Also, you wouldn't even be able to include that on differential. Like, would you get marked down if you put that as a differential, or?
A reactive lymphoid hyperplasia is a differential diagnosis.
Yeah.
I think that would be fair because it is a lymphoid problem.
okay,
What do you think, Diane?
so you could put it in your differential diagnosis, but it wouldn't go in the preferred diagnosis. Yeah, we've got a couple of other questions here. What prognostic features can we mention? Right, the cells around the blood vessels in the stroma, is that a prognostic feature? No, it's not. So prognostic features in lymphoma. So firstly, always there's the stage and the grade, okay? So the grade is related to whether this is a high grade or a low grade lymphoma, which we talked about and is largely related to the intrinsic properties of the cells. Are they big, very active cells? Are they little cells? And the stage, how far it's spread. Now, the prognosis is, of course, dependent on the therapy. So if you remember about lymphomas, the horrible, bad, really, really aggressive lymphomas actually respond really well to therapy. But the low-grade CLL type lymphomas kind of go on forever. So they've got a longer, slower, but inevitable consequence. So the prognosis in lymphomas is slightly complicated by the treatment. So Cam's asked, when you say immunohistochemistry, do you mean fish? And the answer is no, I don't mean fish. So immunohistochemistry is this process where we have antibodies attaching to proteins on cells and then marker antibodies, right? So, and fish is where we have the marker, which is fluorescent, attaching to the DNA. That's the difference between them. Fish is about attaching to DNA, and immunohistochemistry is about attaching to proteins. But sometimes abnormal genes produce abnormal proteins, so we can detect them with immunohistochemistry and by implication say that there is an abnormal gene.
Or absence of gene.
Or an absence of gene, yeah. So that's why we can detect HER2 mutations, yeah, with immunohistochemistry, or BAP1 mutations with immunohistochemistry, or P53 mutations. And then I've got, yeah, and so Amanda says, IHC detects proteins, e.g. CD20 for B cells. So Mars saying, what's the difference between a follicular lymphoma and reactive lymphoid hyperplasia? Okay, so I'm sure you mean probably by histology, because, you know, they've got fundamental biologic differences. So they both have follicles in them, yeah? So reactive lymphoid hyperplasia has those reactive follicles where they're well defined and they have polarity with the centroblasts at one end and the centrocytes at the other, and they have the tingible body macrophages, and they're often in a surround that has multiple cell types present, like plasma cells or eosinophils. Follicular lymphoma, it often has quite a uniform look because you have these round lighter areas, which are the replication areas, in the sea of cells, which are also lymphoma cells. And the follicles don't have polarity, don't have tingible body macrophages, and don't have polarity, don't have tingible body macrophages, and kind of blend into the surroundings. They don't have a definite margin, and the surroundings are all the same cells. So that's how you would tell the difference. Did that clear that up, Mars? It's quite a complicated.
Yeah, I think because I mean when the other ones, they don't have this follicular look at them. They have, both of them have the different type of tissue, basically. I mean, this type of cells. So yeah.
Okay.
Yeah.
So Amanda, would this specimen be expected to be positive for BCL2?
Well, it's not a follicular low lymphoma.
I forgot what Malt Lymphoma immunohistochemistry.
Yeah.
I think this is probably
it's expressed in most low-grade ones, isn't it? Most.
Shall I look it up?
Yeah, we can look it up. Or Lucy, you can look it up for us.
I've got it here. Malt cell should be CD5 positive and BCL2 very positive and cyclin D.
There we go. Yeah. What is the central material in this specimen, Zia asks? So this bit here is a conjunctival stroma. So we've got collagen and blood vessels.
I think it's also called in some of the books substantia propria.
Yeah.
Is that more familiar for you? Multiple words for the same thing.
Okay, that was a really great discussion. Thank you, everybody. I really enjoyed that. So I'm going to just close that screen and go to the next one, which I think should be nice and straightforward because you've seen this several times before. Okay, Amanda, did you want to go through this one or do you want me to carry on?
We can alternate. How about that?
All right, so you'll do this one and I'll do the next one? All right, so I will stop sharing screen and you can nip onto that one.
Try and do this. Okay, I've got it up. And I just need to share screen.
I'll monitor the chat.
Okay, just have to put you all outside of my window so I can see the actual slide itself. Hopefully I can go full screen. Oh, okay, is that good for you?
Yeah.
Right. Are there any questions, first of all, about what you found difficult? So I can just address that directly. You can ask directly or put them in the chat, whatever. Otherwise, I'll just start describing. This is a pupil optic nerve section of an enucleated globe. And I can see in the posterior globe, there is a large mass lesion situated in across the equator. It appears to be arising and expanding the choroid. It has a dome shape. Anteriorly, it appears to involve the ciliary body, because I can tell there's a little ciliary process here, looks like the ones on the other side. And the ora serrata is probably around here. So it extends from posterior to the equator, anterior to the ora serrata to involve the ciliary body. At this low power, I can see it's a mass lesion, it's a tumor, it is producing pigment. The other pattern that I can see at low power is it's got this perivascular sleeve pattern, which we can see in retinoblastoma, but that's not specific for the tumor type. It just tells us that there's the blood vessel inside and a oxygenated rim of tumor around it. I can also tell that the pink areas are areas of necrotic tumor. Before I go too close, the other things I would like to look at are, I would see if there is involvement of the anterior chamber, which I can't see in this angle, but this angle looks clear. The sclera, there is no extrascleral invasion of the tumor, and the optic nerve also looks clear. The lens has become displaced and has just fallen into this part of the posterior part of the globe. So at this low power, I think this isn't going to be a choroidal melanoma. And now I would like to find prognostic factors, including cell morphology. So I'll zoom up to a part that's not too pigmented so I can see it properly. That would be my tip. I want to see if it's spindled epithelioid or mixed. There we go. Well, I can see at least spindled cells here, and there may be epithelioid cells as well. Other features, other prognostic features, I did the anterior chamber. Size, I forgot size at low power. So the basal diameter, considering this is a whole globe, it's probably about 10 millimeters across and maybe 5 millimeters in height. So this would be in most likely the moderate size category. So my differential diagnosis, in an adult with a pigmented uveal malignant tumor, I don't actually have a differential diagnosis. I think this is a choroidal melanoma with no differential diagnosis. If there was no pigment, I would consider it could be a metastatic tumor, but this is making its own pigment. So I think I don't have a differential diagnosis. Special stains that I would do, I don't need to do anything to prove that it's a melanoma because I know it's making pigment, so it's already shown its differentiation to me. But I would do prognostic markers. I would do immunohistochemistry for BAP1, and if it's lost, that's a poor prognostic factor because it probably indicates loss of chromosome 3, and I would do FISH on the paraffin section for 3, 6, and 8. That's how I'd answer that question using our structure. And the clinical presentation, well, you can figure that out. It's a dome-shaped lesion there in the posterior part of the globe. Questions?
Could anyone see any perineural or perivascular invasion here?
Right. Perineural invasion.
I didn't think it was present, and I didn't think there was invasion of the optic nerve either.
No. So yes, of course, the biggest nerve around is here, and not involved, which we can see at low power. And vascular invasion, we would look out here in the sclera, in the scleral veins.
We thought there was intrascleral invasion.
In a little bit, yes, in the inner 1/3 perhaps, Lucy? What do you think, Diane?
Yeah, I think it is extending in there. 1/3 of the way through, but not all the way through. Yeah,
not all the way, so not extrascleral. So intrascleral, that doesn't put it into a poor prognostic category. It's still contained within the globe. I can't see any intravascular invasion. Did anybody else see any? Or any other questions? I can't see that. When you're sharing screen, the chat window goes away, so I can't see it.
Sorry, I should pop up the chat here.
How's blood vessels that?
Okay. All right. So right, for flow cytometry on solid tissue, do you mush it up first?
With regard to this tumor, or with regard to?
Oh, that was a hangover from the last one.
Oh, all right. Sorry. We send it down to flow cytometry and they disaggregate the tissue themselves.
Right.
All right. Would you say the differential diagnosis ciliary body melanoma given the different prognosis? So uveal versus ciliary body.
Okay.
So
So
choroidal versus ciliary body.
So that's the site, right? So they have not included this important part at the bottom here, unfortunately.
No. So if it was involving the ciliary body, you definitely mention it as a prognostic feature.
And we think it is because here's ciliary processes and oris serrata is probably here. So which marks the end of the pars plana of the ciliary body. So ciliary body, pars plana, oris serrata. Oris serrata. So if I draw the line about here, most of it's in the choroid, but certainly the ciliary body is involved. So that's what we determined at low power.
Okay, and can we see the spindle and epithelioid cells close up? And is there an extracellular matrix pattern?
I usually do extracellular matrix with a PAS stain. What do you do, Diane?
I just look for it.
You just look.
But in the solidy bits is where I often look for it, because you can actually see their things. This is quite thickly cut, isn't it?
Yes, so we've got the spindled cell component.
So I don't think there's epithelioid cells here.
Yeah. Just trying to find some, if there are any. And like in this, I'm trying to find more solid bits to see if it has that extra vascular loop pattern or not, but I'm having a rendering problem.
Yeah.
It has some just underneath the ciliary body. Definitely has that.
In the solid bit, yeah.
And the solid bit underneath the ciliary body, it definitely has it.
So more
Maybe in
anterior.
here.
More anteriorly.
There, in that bit, yeah.
Oh, we can't get any more anterior than this, so it's slowly rendering. It's coming in from the left. Isn't that interesting?
Probably like the election, coming in from the left.
There we go. One electrode at a time.
Yeah.
It's actually cut a bit thick.
It is.
Yeah, possibly here. It's hard to tell because of the amount of pigment here.
I wasn't certain I saw any here.
Yeah.
Yeah.
I think I'd like a thinner section and to do a PAS, but you're asking the right sort of questions and you're looking in the right sort of area.
That's
Is that
good.
why it's so dark that it's thick?
Yeah.
Cut? Yep.
Just been cut thick.
Yes, it's not just because of the melanin, because of this, the amelanotic part over here is also difficult to see into. Eyes are pretty difficult to cut because they're such large things, and there we go. Still spindled actually, so.
But I think your thing's too spindled. Right, can you tell primary choroidal melanoma versus secondary from distant metastatic?
Well, that's a really good question because they'd both be melanomas, wouldn't they?
They would be.
Now, what I think, what does the team think? I think the clue might be not morphologic features, but something else. What's one of the genetic features that's different between cutaneous melanomas and uveal melanomas? And we discussed this last time about what oncologists ask for.
Is it BRAF?
So if there was BRAF present in this tumor, what would that tell you?
Probably cutaneous or conjunctival.
Yeah, and NRAS as well, Chris has written. Yeah. And the other thing is that I think melanomas from the skin don't have the characteristic monosomy 3
Yes.
and 6 and 8 changes. So the genetics will be different.
The genetics. I don't think we'll be able to tell. Would you be able to tell if this was met to the choroid from a cutaneous melanoma?
No.
No.
I would have thought that the clinical scenario would have been helpful.
You should have spotted it, yes, clinically, or the patient should have told you.
Yeah, usually by the time it would have got to the eye, it would have got to the brain.
Yeah, because that's what it's doing. Okay, good.
My turn to do the next one?
Yes.
All right.
Sorry,
And then we'll take a short break to get our energy back again.
Sorry, can I just quickly answer that last?
Juliana's got a question.
Yeah, I just was wondering, did you call that a spindle B melanoma?
Well, the current classification, the choices are spindled, epithelioid, or mixed. That's it.
Okay. Okay, so you don't have to break it down. Okay, that's fine. Thank you.
All right, last before we take a re-energizing break. So we've got this piece of tissue and there's no really normal tissue present, except maybe just out here where we've got some loose connective tissue with inflammatory cells in it. Oh, that's a great pixelated thing.
That's right. You can type in your questions that you had while you were doing it by prior also, so that we can address that as well.
Okay. So I think this is the normal tissue. We've got little bits of normal residual fat, and we've got some loose connective tissue with blood vessels in it. So I think that this, knowing that we're doing eye pathology, that this is an orbital biopsy. How did people find this one?
Questions?
difficult. It is difficult. So it's an orbital biopsy, and we've got this great hulking lump of stuff with a bit of normal orbital tissue here. And we can see that it's got inflammation in it, can't we? And we can confirm that it has got inflammation when we go and have a look at these inflammatory areas here. So it's a predominantly lymphocytic infiltration. And then there's a bunch of fibrosis out here. Yeah, this is all collagen. The collagen in places is wrapping itself around blood vessels. Yeah, but really it's got this kind of almost styrofoam look. By styrofoam we mean kind of spindle, kind of just spinning out kind of look. So we've got an orbital biopsy that is showing large amounts of fibrosis and chronic inflammation. So we've got to decide at the moment first, is it a tumour in the orbit or is it not a tumour? And the presence of inflammation and fibrosis suggests to me that this is not a tumour. So when you sent it into the lab, you might have been thinking it was a lymphoma and done a lymphoma protocol. However, when me, the pathologist, looks at this, the degree of fibrosis, plus just down here, we might have some reactive lymphoid follicles. Maybe not. There's a hint of one.
Almost.
But yeah, that's a little reactive lymphoid follicle. Just here would suggest that this is a reactive process. So the next thing you need to do is describe it. So we've got patchy reactive lymphoid inflammation together with dense sclerosing collagen in a no particular pattern. We often use a patternless pattern, but there is a somewhat characteristic wrapping around little blood vessels here. So then we need to apply our clinical knowledge to generate the differential diagnosis. And what is the differential diagnosis of massive sclerosis in the orbit? So in common things that I'm thinking of, it would be idiopathic orbital fibrosis, IgG4 disease, not thyroid eye disease, because thyroid eye disease affects the muscles and spares the orbital fat from fibrosis. The orbital fat undergoes a different change, but it doesn't become densely fibrotic like this. Yeah, so what other diagnoses would there be?
Let's think of some other ones. So Lucy had it in her slides, pink deposits of material that could occur in the extracellular space.
Hyaline.
What about amyloid as well?
Yes, it could be amyloid, couldn't it?
Yeah, that's right. Yeah, Nishan and Cam are onto it.
So apart from IgG4 disease, which an idiopathic fibrosing.
Yeah, IOI.
IOI.
Our eye surgeons really don't like the word orbital pseudotumor.
So we've all gone off orbital. It's idiopathic orbital inflammation, isn't it?
Yes, IOI.
Idiopathic orbital inflammation.
Although the AAO book calls it something slightly different. The AAO book calls it non-specific orbital inflammation, NSOI.
So the other thing that I initially thought is, could it be sarcoid, because that's associated with lots of fibrosis, but I didn't think that there were any granulomas. So am I still sharing my screen here?
Yep, we're looking at Eagle Eye book.
Eagle, yep. So idiopathic orbital inflammation is what we think it is. Yeah.
And often, and so you would give steroids typically for this, right? Do you think this one would reduce in size?
I don't think so, because the fibrosis is very well set in. It would shrink a tiny bit, but.
How would we know if it's IgG4?
Yeah, so We would know it was IgG4 in several ways. So the first thing is that before you sent the biopsy, you probably had a differential diagnosis, which was quite wide and included things like, lymphoma and sarcode and IOI and IgG4 disease, and you would have done the workup for those. And so IgG4 disease often has an increased IgG4 in the blood, but down the microscope, we would stain for IgG4.
Did you see many plasma cells in here?
I did not, and plasma cells are associated with IgG4 disease.
Because that's what makes it. That's what makes the immunoglobulin, the plasma cells.
That's right, and it's the plasma cells expressing IgG4 that show it. So there's no granulomas, there's no plasma cells, just seas of lymphocytes. So I thought that my preferred diagnosis would be IOI.
Yep. And there's some good questions coming up here. Is the sclerosing type of IOI the same as IgG4?
IgG4 is, by some people, thought to be a subset of it. But in reality, I disagree with that, because I think that IgG4 is a systemic disease and is distinct.
So IgG4 related disease is often what we call it, and there will often be other organs and or parts involved. Did you see any lacrimal gland here? Because IgG4 often goes to lacrimal gland, doesn't it?
It does, and I didn't see any lacrimal gland here.
So that would be something to look for.
Yeah, I'm just looking to see if I missed it in my low power scan.
And so Lucy's asked, in the special tests, would you say do immunohistochemistry for IgG4?
Yes.
Yes, that's a very good thing to do. And would you say rule out infection by doing microbiology testing?
I don't think so. I think that once I've seen this, it would be a diagnosis of IOI or IgG4. However, if you were asked about sending an orbital specimen to the lab, and you didn't know what it was, then certainly an infection, particularly a slow infection, like maybe a fungal infection or a slow bacterial infection, would definitely mean that when sending samples, you would consider sending a sample to the microlab.
Because that's inflammatory mass. What would you do? That's the inflammatory mass is the bigger category.
Yeah.
And then Nishan says, would you do a Congo red for amyloidosis?
Yep.
Yep.
Just in case, it's cheap and easy and you don't want to miss it.
Good.
Right, I will stop sharing that one. Do we think it's time, so we've been here like an hour, time to get up and walk around for a bit?
I think so.
All right, we've done 3 slides, we're doing well.
So we'll stop recording and come back in 5 minutes?
Yep, stop recording, come back in 5 minutes.
At 12:05 our time.